So we will continue with the enzymes of genetic engineering. We had finished about DNA polymerases, and today we will look up into the next batch of enzymes, which are the DNA ligases. I will be sharing my screen soon. Okay. So, before the DNA ligases, we must talk about reverse transcriptase. Reverse transcriptase is an RNA-dependent DNA polymerase. So you had read about DNA-dependent polymerases, and DNA-dependent polymerases, what they do, they synthesize on one DNA strand, another DNA strand. If one DNA strand is in the 5' to 3' direction, the second DNA strand is synthesized in the 3' to 5' direction. So this is it uses DNA as a template, but reverse transcriptase, it makes DNA from RNA. So if you have a single-stranded mRNA, that is converted into a DNA. Single-stranded DNA is converted, and that is known as cDNA or complementary DNA. So it produces a DNA strand from an RNA template. So this has no exonuclease activity. So there is no digestion from the ends. So this enzyme is basically used for copying messenger RNA molecules in the preparation of cDNA. So one mRNA is converted into a cDNA. And this cDNA is used in PCR, basically in PCR for cloning. And reverse transcriptase, it is a key enzyme in the generation of cDNA. And it is an RNA-dependent DNA polymerase which produces a DNA copy of an mRNA molecule. When I will be talking about uh, cDNA preparation, then we will RT-PCR cDNA preparation. We will talk in details on this. Then you have enzymes that modify the ends of the DNA molecule. So these are the enzymes which are modifying somewhere in between. Although exonucleases, they are digesting from the ends, but basically this is creating another. But then you have the DNA, the 3' and the 5' ends, which modify. There is the enzyme alkaline phosphatase. There is this enzyme alkaline phosphatase. Then polynucleotide kinase, specifically T4 polynucleotide kinase. Alkaline phosphatase, what it does, it removes the phosphate from the 5' on the 5' end. So 5' triple 3 phosphates are there, it removes that phosphate. So one single phosphate is there, that is removed. Result, there becomes an OH group. And because if it is removed, it will, once the phosphate is removed, it will not ligate. We will talk about this ligation a bit later. And polynucleotide kinase. So opposite of phosphatase is kinase. Kinase adds a phosphate. So if there is no phosphate, it will add a phosphate. They end to the end, termini, to the end of the DNA molecule and provide important functions that are used in a variety of ways. The phosphatase and kinase enzymes, as the name suggests, are involved in the removal or addition of phosphate groups respectively. You normally used to use calf intestinal phosphatase. It removes phosphate groups from the 5' ends of the DNA. So this enzyme is used to prevent unwanted ligation of the DNA molecules, which can be a problem in certain cloning procedure. So what the function of DNA ligase is to join DNA. But if there is no phosphate on the 5' end and the 3' end also doesn't have phosphate, it has got OH. So two OH molecules, they cannot form a phosphodiester bond to make a DNA molecule. Phosphatase is OPO. There is no P for, so therefore the two OHs will not work properly. So this is important. Some problems happen when if there is this phosphate. Suppose you have digested with a restriction enzyme, and you want to put an some insert inside your vector. So problem will come when the moment you add DNA ligase, the vector will self-religate before the phosphate can enter inside. So this is prevented by removing the 5' phosphate. Then you have terminal transferase. It repeatedly adds nucleotide to available 3' terminus. So it will be keeping on adding nucleotides at the 3' terminus randomly. Or you have a terminal transfer T T T T T T. It will keep on adding. So this enzyme is mainly used to add homopolymer tails to DNA molecules prior to the construction of recombinants. We will look at cloning, but know that name of this enzyme. So phosphatases, kinases, transferases. You know what are the functions of these enzymes. Before we go to the cloning part, we must know what are these enzymes. Then DNA ligase. What it does? It creates a phosphodiester bond. A phosphate is needed, and the help of ATP, two DNA molecules are joined. Cut DNA molecules are joined. DNA ligase. In genetic engineering, it is used to seal discontinuities in the sugar phosphate chains that arise when recombinant DNA is made by joining DNA molecules from different sources. So it's like a glue, molecular glue. This function is crucial to the success of many experiments, and DNA ligase is therefore a key enzyme in genetic engineering. So most we use T4 DNA ligase. Then another important enzyme is the polymerase amplification. We do is the polymerase chain reaction (PCR). So ligases. So this is a DNA fragment. You have a sticky end. This is the plasmid DNA. This is the DNA fragment. Although they will this will come and the join over here, this blue will come on top of it. G opposite of C, A opposite of T, and this is the same end, but still these two will not join because they are separate. They will come close, but they will not join. This joining is done by DNA ligase. The T4 DNA catalyzes the formation of phosphodiester bond between 5' phosphate and 3' OH in duplex DNA or RNA. This repairs single-stranded nicks in duplex DNA, RNA, and DNA-RNA hybrids. It also joins DNA fragments with either cohesive or blunt termini, but it has no activity on single-stranded nucleic acid. It needs two strands. It requires ATP as cofactor. Also, you have T4 RNA ligase. They will attach RNA. So there are the modifying enzymes which represent the cutting and joining functions in DNA manipulation and genetic engineering. So what is an enzyme? You know that enzymes are proteins and a certain class of RNA, ribozymes, which enhance the rate of a thermodynamically feasible reaction are not permanently altered in the process. So what do enzymes do? They make a substrate into a product and they themselves are not changed. So mostly they are proteins, but there are also RNA enzymes like ribozymes. So then you have the molecular scissors or the restriction enzymes. They are the molecular scissors. So a restriction enzyme is an enzyme that cuts double-stranded or single-stranded DNA at specific nucleotide sequences known as restriction sites. So E. coli, whenever it sees the sequence GAATTC in a double-stranded part, it will cut that region. So property of restriction enzyme is they break the phosphodiester bonds that link adjacent nucleotides. So পাশাপাশি phosphodiester bond is formed, it is going to be cut from the center. So how do restriction enzymes work? The restriction enzymes, they recognize a specific sequence of nucleotides. So as I told you, they recognize specific nucleotide sequences. And this cut can produce sticky ends. This is a sticky end. Ekdike there is an overhang. And this is a blunt end. There is no overhang. So this is a blunt end. And this is a sticky end. একটা overhang রয়েছে. Clear what is blunt end, sticky end?
Sir.
Okay. So these blunt end fragments can be joined to any other DNA fragments with blunt end. One blunt end can be joined with another blunt end. Enzymes are useful for certain types of DNA cloning experiment. We will study this when it's used for cloning. It will create blunt ends. Sticky ends. DNA fragments with complementary sticky ends can be combined to create new molecules which allow the creation and manipulation of DNA sequence from different sources. So, if you digest with only with EcoR1, it will only join with another EcoR1 site. It will not join with any other site. But blunt end, any blunt end, it will join. While recognition sequences vary widely, the lengths may be between 4 and 8 nucleotides, and many of them are palindromic. Madam is palindromic. From whichever direction you go, you read madam from the front to the end or end from the front, it looks the same. That is palindromic. And the recognition sequence is 4 nucleotides to 8 nucleotides. Most of them are 6 nucleotides. So GAATTC. So the other way around, you read GAATTC, the complementary. So this is a palindrome. This is the EcoR1 recognition site. So you have seen what palindrome is. Star effect porte hobe na. And I have talked about EcoR1 before also in the class. So we have 3 types of restriction enzymes. Normally we use type 3 mostly. EcoR1 is type 3. But there are other 2 types, that is type 1 and type 2. Type 1, they are capable of both restriction and modification activities. They need SAM, S-adenosine methionine, SAM, and ATP. They cleave DNA at random length from recognition site. They don't have specific site inside. Like GAATTC, it will always cut after the G. Type 2, their recognition are usually undivided and are palindromic, 4 to 8 nucleotides in length. They recognize and cleave DNA at the same site. They do not use ATP for their activities. They usually require magnesium as cofactor. So the type 3, they cut 20 to 30 base pairs after the recognition site, not inside the cut. So they require adenosine, adomate and ATP cofactors. They need it additionally. So these are some restriction enzymes. This we will talk about these later. In the next class, we will talk about genetic engineering vectors. This I will take in your classroom. We will talk about vectors, plasmid DNAs. So any questions so far?
Sir, adomate কি?
Huh?
Adomate?
Adenosine, methyl adenosine. Methyl
Yes, sir.
adenosine. It's a compound. There is one S methyl adenosine, then one adenine base to this CH3 is added, methyl. Clear?
Yes, sir.
Okay. Okay, Tata.
Thank you, sir.
You're welcome.